So our next speaker is Megan McCain from the University of Southern California, and she's going to actually build on the iPSC theme, taking it into a slightly a more complex system and ultimately look at characterizing a disease. So the title of her presentation is Modeling Barth Syndrome Associated Cardiomyopathy with Human iPSCs and Microphysiological Systems.
Great. Thank you, Brian, for the introduction, and thank you to the organizers for inviting me. I've certainly learned a lot at this meeting. So I made my title a little bit more broad so I can kind of introduce you to the field of microphysiological systems and how we use them in my lab to use for building human cardiac disease models. All right, so before I get started, just wanted to acknowledge my group. So I've been at University of Southern California about 3.5 years, so these are my graduate students. Some of the work I'm going to talk to you about I did when I was still a trainee in Kip Parker's lab at Harvard, so I wanted to acknowledge him as well as funding sources throughout the years. All right, so my lab primarily studies the heart. So like all biological systems, there's really key structure-function relationships that scan multiple spatial scales that are critical for the function of the heart. So as we all know, the key function of the heart is to contract and pump blood. The main tissue that achieves that contractility is the myocardium. So this is the striated muscle tissue within the left and right ventricle. If you zoom into a histology slice of human cardiac tissue, it's packed with rectangular cardiac myocytes. So the shape of these cells is really critical for the function of the heart because they're long, elongated, all the myofibrils will be aligned in the same direction. So as the sarcomeres contract, they're all working in the same direction, and you get kind of optimal efficiency. The alignment of the tissue is also critical for the same reason. So kind of the cell tissue architecture is something that we focus on quite a bit. If you zoom in even more, you can see here in the schematic, again, myofibrils elongating parallel to the length of the cell. Cardiac myocytes are also coupled by very specialized cell-cell junctions that ensure mechanical and electrical continuity. So that's why the ventricle acts essentially as a syncytium, even though it's individual cells. Myocytes are also coupled directly to the extracellular matrix, which is going to provide the resistance for the cells as they contract. So if you think about fibrosis and different disease states, that resistance and the load on the cells is going to increase. So when we think about the function of the healthy heart, how the function can decline in certain diseases, we really want to consider all these different spatial scales and how cell tissue architecture, the microenvironment, how that can impact disease progression. All right, so coming at this also from kind of an engineering perspective, we need to be able to deconstruct heart disease so that we can build simplified models and then add complexity as we go. So if you think about heart disease, there we go. The most common causes are acquired, so these are things like myocardial infarctions where you have blockage in a coronary artery causes necrosis in downstream cardiac myocytes. Because myocytes are non-regenerative, they're replaced with fibroblasts, scar tissue, which first of all is not going to be muscular, so that's going to reduce the contractility of the organ. And then it's also going to increase the mechanical load on the surviving myocytes. So as a result, you get pathological hypertrophy that often progresses to dilation and heart failure. So again, remodeling of the microenvironment is really a key feature of this type of disease modeling. There's also certainly examples of inherited cardiomyopathies. So the example here is hypertrophic cardiomyopathy, where all the numbers here point to different proteins that are known to have mutations that can cause this disease. So hypertrophic cardiomyopathy and a lot of the inherited diseases tend to affect patients at younger ages. So these are the diseases where if you hear young people having heart attacks at marathons, things like that, right, they're usually inherited conditions. And certainly all diseases essentially are some combination of both, right? And kind of understanding how both of these factors can contribute to disease progression is important. So independent of the root cause, we still see some common features in how the tissue can remodel in different disease states. So here's a slice of healthy cardiac tissue. Here's one from heart with hypertrophic cardiomyopathy, but we see some similar features independent kind of the root cause. So some of the things we see are cellular remodeling, so changes in cell shape, changes in tissue architecture, changes in the proportion of fibroblasts to myocytes, changes in the extracellular matrix, so this can be composition as well as elastic modulus. So healthy tissue has a elastic modulus of about 10 kilopascals. This goes up to over 50 in fibrotic conditions. And then changes in mechanical loading, either due to stiffness or changes in afterload or some combination of both. And then kind of on the phenotype side, things that are commonly seen are remodeling of the ventricle, arrhythmias, inefficient metabolism, contractile deficits. All these things are interrelated in some ways, right? So the issue is a lot of the existing cardiac models have some key limitations where we think we could add iPS cardiomyocytes combined with the microphysiological systems that I'll describe to overcome and kind of complement some of the limitations. So animal models, as we've learned about at this meeting, are great for studying kind of native physiology in an intact organism, right? So we can get all the organ-organ interactions, what happens in an intact organism. And in vitro systems are probably never going to be able to capture all that complexity, right? But the issue with animals, of course, they're non-human. So again, looking at specific genetic diseases is challenging in animal models. Limited control, so it's difficult especially to control those microenvironmental parameters in animal models to robustly study them. Limited access in terms of being able to monitor cell and tissue remodeling in real time is challenging in animal models for the heart. And then, of course, challenges in terms of throughput and cost from kind of a practical perspective. On the other end of the spectrum, conventional cell culture, now we can get experimental control, we can use human cells due to a lot of the iPS technology, and we get benefits in terms of throughput and cost. But of course, we're losing all the native tissue structure that I described to you that's really essential for the function of the heart. We're lacking physical cues, so we're growing cells on a plastic petri dish, right, where we know that the elastic modulus of the heart is much lower than that, and that's important, again, for myofibril development. And then limited functional output. So for cardiac myocytes, we care about contractility as kind of the key output, and it's difficult to measure that using just conventional tissue culture plastic. All right, so coming at this from kind of an engineering perspective, what are the key design parameters that we would like to engineer in a disease model to kind of complement the existing model systems? So in terms of inputs, we want to be able to tune cell demographics, cell genotype, cell tissue architecture, and multiple properties of the extracellular matrix, as well as mechanical forces. Because again, those are kind of key features we see remodeling in a lot of different forms of disease. On the output side, we of course want to be able to look at gene-protein expression, kind of standard molecular biology, but we also want to be able to quantify cell and tissue morphology and more of these functional metrics as well in our models. And then from a practical perspective, we want our technologies to be modular, so we can kind of pick and choose from this list, what parameters are really important for us to control and measure in our specific question. We want them to be scalable, so they can ultimately be adapted for drug screening, for example. Of course, robust and quantitative, all these reproducibility issues that we've been discussing here. And then human relevance and patient specificity is another key feature for these systems. All right, so when I was a trainee, I worked in Kip Harker's lab at Harvard. He's done a lot in the organ-on-chip space, and one of the platforms developed from his lab while I was there is called muscular thin films. So again, kind of our design parameters here are to use human cells, as I'll describe, but ultimately this platform is for measuring contractility, so we can get the key functional output of the tissue. So the way this platform works, we start with a glass cover slip, cover it with masking tape. We then use a laser engraver. So some of you may have seen these at craft fairs or places like that. It's for, you can laser engrave your iPhone or make Christmas ornaments, so a very kind of commercial off-the-shelf system. We can cut these small windows out, coat this with a temperature-sensitive polymer, so it's called polyanisopropyl acrylamide. Essentially, this polymer is solid when the cells, when it's at 37 Celsius, so within the incubator, and then at room temperature, it will melt away. So it's kind of a switch that we control when this polymer will dissolve. We then coat that with PDMS. This is a silicone elastomer. It's clear, non-toxic, flexible, FDA approved, widely used for a lot of kind of these engineered system studies. We then use our laser engraver again to cut these cantilevers, so they remain anchored at their base, but the other sides are free. And then we do a technique called microcontact printing. So this is essentially rubber ink stamping, but on the micron scale. So we design different patterns in CAD software, transfer them onto a wafer in a clean room using the same technology from the computer chip industry. Then we can mold PDMS stamps. So this is the same silicone rubber that we can get these patterns transferred onto as a stamp. We then coat it with matrix protein. We usually use fibronectin, but it's compatible with laminin, Matrigel, other types of matrix proteins as well. Incubate it for an hour, dry the surface, and invert it just as you would a rubber stamp. This transfers the fibronectin in a defined pattern, and then we can seed our cardiac myocytes, and they adopt whatever pattern that you lay down. So now we have a kind of tunable way of controlling tissue architecture, and then ultimately the next step of this is letting the cells grow for a few days, form a confluent tissue. We then move the constructs to a room temperature microscope, so that will dissolve away this temperature-sensitive layer, and the films will actually contract now because they're free, except for at their base. So the way this experiment looks in real time is shown here. So now we have a layer of the PDMS, a layer of the cardiac myocytes. We do have an electrode here to control their pacing rate. And we can monitor this from above, and based on the curvature of the film, we can now calculate stress. So now we have a relatively sensitive assay to measure the key function of these cells. Okay, so a lot of our platforms we develop with neonatal rat cardiac myocytes, which is what I showed you on the previous slide, but of course we want to be able to do human disease modeling. So this is where the IPS technology that Paul described, except we actually start usually with dermal fibroblasts, so skin fibroblasts from a patient, reprogram them to iPS cells, and then differentiate to cardiomyocytes. So we collaborated with Professor William Poo. He's a clinician at Children's Hospital in Boston. He had collected cells from patients with Barth syndrome. This is an inherited cardiomyopathy. It's due to a mutation in taffatsin or TAZ. This is an enzyme that processes a key lipid for the mitochondrial membrane in striated muscle. So patients with this disease tend to need a heart transplant by adolescence. Again, not a lot of therapeutic approaches, not great animal models for this disease. So he had collected skin fibroblasts in their lab. They reprogrammed them to induce pluripotent stem cells, differentiated into cardiomyocytes, did some studies to validate that the cells were recapitulating the phenotype that they're seeing in the patient in terms of cardiolipin production, which is shown here, and also ATP generation. So because the mitochondria are not forming properly in these cells, ATP generation is also affected. And another important thing here is we had 3 wild-type patients and 2 patients with Barth syndrome that had different types of mutations. So that's another huge advantage of this technology is we can get at kind of allele-specific effects and functional outputs. So ultimately, we wanted to look at the contractile phenotype of these cells and identifying ways of potentially rescuing it. So now, these are the same muscular thin films I showed you before, but now with human-induced pluripotent stem cell-derived cardiomyocytes from healthy patients and patients with Barth syndrome. So this is actually also a movie, but the cells were so weak that they barely moved the film. So a few experiments here and there, we'd see a little bit of twitching, but ultimately a pretty drastic decline in contractility compared to the wild type. We could also show that we could recover the phenotype by re-expressing TAZ, so that's showing that the disease is really caused directly by the mutation in the TAZ gene. All right, and so now this kind of gives us a platform, right, to do disease modeling, hypothesis-driven research, ultimately drug screening. So it's kind of a proof of concept study. We showed that reactive oxygen species were built up in patients with Barth syndrome, again, likely due to the defects in the mitochondrial membrane. So we were able to add mitotempo. This is a scavenger for ROS. Let me see if I can play the movie again. Here we go. And recover some of the contractility after the cells had a few days to kind of recover and rebuild their myofibril network. So this, again, is kind of a great platform for being able to do these types of mechanistic studies in a human-relevant platform. We also did CRISPR mutated wild-type lines as isogenic controls, again, to really validate that this is due to mutations in TAZ. Some other compounds we looked at were linoleic acid, so this is a fatty acid precursor to cardiolipin. So the idea is if you load the cell up with the precursor, maybe you can get enough to recover the function. We also saw some recovery there. Again, using this as a platform for identifying therapeutic approaches. So it's certainly not high throughput. This I would classify as kind of a medium throughput platform, but it kind of gives you a nice bridge between the high throughput cell culture and multi-well plate format before going to your animal model, or kind of complementing the data that you do get from the animal models. Another thing that we use this microcontact printing approach for is to micropattern small islands of fibronectin, where then you can get isolated single cardiomyocytes. And what's great about this is now you can analyze and quantify cell morphology in a very robust system. So when cardiomyocytes lay down on these fibronectin islands and fill in the shape, they form structures that are very repeatable. So now you can do more quantitative statistics and analysis of cellular structures, again, towards this idea of reproducibility. All right, so I'm going to start speeding up a little bit. So in my own lab, this kind of spurred our interest in interactions between mitochondria and sarcomeres. So the Barth syndrome story is really due to a mutation causing an inherited cardiomyopathy. So what about the microenvironment, and how do changes in the microenvironment also affect metabolism that could potentially synergize or interact with mutations? So one area that we're interested in is looking at how things like the microenvironment in terms of the matrix stiffness, the alignment of the tissue, how the cells are organized, how do those things affect mitochondria, and is there some pathogenic effects of these features that are causing metabolic defects? So if you look at a slice of healthy cardiac tissue, it actually forms pretty beautiful structure in terms of elongated myofibrils with mitochondria really crammed inside. So as you can imagine, as the cell's shape maybe remodels due to different changes in the microenvironment, that might have a critical impact on mitochondrial function as well. So the problem is the kind of key way of studying oxygen consumption is with this seahorse extracellular flux analyzer. I'm sure a lot of you are familiar with this. You add different components to block different channels within mitochondria to pull out some of these factors. The limitation is you have to use their cell culture plate. So a lot of the techniques I showed you, like microcontact printing, we usually do that on a larger format. So it's very difficult to do those types of assays within these multi-well plates. So to get around this, what my student Davi did is basically do all the tissue engineering outside of the plate, and then as the very last step, transfer the engineered substrates into the well plate so that we can still control features in the microenvironment and measure using, in this case, the seahorse machine, but you can imagine any type of plate reader assay this would be compatible with. So the way that we do this, I'll start here. You start with a glass cover slip. We add our temperature-sensitive polymer again. This is the switch that allows us to release surfaces easily. We coat it with PDMS. This is our soft, our silicone elastomer. And there's actually different varieties of PDMS that have different stiffness. So if you want to mimic the stiffness of a developing heart or an adult heart or a fibrotic heart, just kind of depending on the recipe of PDMS you use, you can get that tunability. We then use the same laser engraver now to cut individual circles that are the same size as the bottom of the well plate. We do the same microcontact printing I showed you before, and now we can peel up these discs, place them into the bottom of the well plate, seed our cells, and be able to now control tissue architecture and matrix stiffness within these well plates. So these are now images of tissues taken within the plates. This is just validation showing you that yes, this works, so we can control matrix stiffness, we can control tissue alignment, again, all within the plate, and we're seeing nice sarcomere formation, aligned tissues versus isotropic tissues here. All right, so in terms of actual function, so how do these parameters in the microenvironment that do have relevance to disease affect ultimately oxygen consumption rate? So we did these experiments with Roberta Gottlieb at Cedars-Sinai. She runs a big mitochondrial core there. They're really instrumental for getting and analyzing this data. So here, this is isotropic tissues, and these are the aligned tissues, and the different colors represent different matrix elasticities or stiffnesses. So you can see in both isotropic and aligned tissues, there does seem to be a response based on stiffness, which makes sense as the cells are having to contract against a higher load. You'd imagine that their oxygen consumption rate, their metabolism is going to increase to generate enough stress to adapt to that mechanical condition. What was interesting is if you look now here, this is maximum respiration in aligned tissues versus isotropic tissues. We saw very little difference on the isotropic tissues, but in the aligned tissues, we see the stiffer tissue consuming more oxygen, so kind of the maximum rate that these cells can behave at. So that suggests some kind of interesting interactions between the mechanical environment, the alignment of the tissue, and giving us some insight into how remodeling of the microenvironment could affect these parameters. All right, so I'm running low on time. This basically summarizes what I just said, but now in kind of bar graph form. So this is my last slide. So this is kind of the framework of my lab and the type of work that we do. So again, this kind of design, build, test algorithm. So we do a lot of work looking through the literature, working with collaborators who have disease expertise, understanding what are the key features in the microenvironment that are changing in healthy and disease, both cardiac. We also do work with skeletal muscle that I don't have time to talk about today. Then we use a variety of different tools, so microfabrication techniques, biomaterials, human relevant cell types such as iPS cardiomyocytes. Do a lot with assay development, either developing new assays to measure contractility or kind of adapting existing assays to work with our engineering tools. Our main application is disease modeling, so looking both at fibrosis as kind of a disease stimulus, and then also working with collaborators, so to get patient-relevant cells. So we work with one group doing Duchenne muscular dystrophy, another group studying ALS. So we're able to get patient-derived cells by working with these kind of clinical and medical school collaborators. We also do some work with microfluidics, so trying to miniaturize these platforms so that we can, again, increase scalability and be able to use them for kind of medium throughput drug screening. And then ultimately some of what we do is relevant also to cell therapy and kind of improving the phenotype of cardiomyocytes. So that's it. I guess we'll take questions at the end of the panel. So thank you.
Thank you, Megan.